Today I am going to talk to you in detail about the structure of the cell and the functions of the various organelles. I will discuss in detail the differences between prokaryotic and eukaryotic cells. The word cell comes from the Latin word cella, meaning small room. A cell is the basic structural, functional, and biological unit of all known organisms. It is the smallest unit of life. The study of cells is called cell biology or cytology. The number of cells in plants and animals varies from species to species. Humans contain around 40 trillion cells. The human brain contains about 80 billion cells. Cells emerged on Earth at least 3.5 billion years ago. The smallest cell is mycoplasma, also called pneumonia-like organisms. It is about 10 micrometers in size. The largest cell is the egg cell of the bird ostrich. The longest cell is the nerve cell. A unicellular green alga called Acetabularia has the largest plant cell, which can reach up to 10 centimeters in height. Cells were discovered by Robert Hooke in 1665 in cord tissue. He took thin slices of cord and examined under a crude microscope that he developed. Actually, he was examining dead cells. His observations were published in Micrographia. The first one to examine living cell was Leeuwenhoek. He discovered blood cells, microscopic nematodes, sperms, etc. In 1831, Robert Brown discovered nucleus in orchid root cells. In 1839, Purkinje coined the term protoplasm for the fluid substance of the cell. In 1838, Schleiden, a German botanist, made extensive microscopic observations of plant tissues and described them as being composed of cells. In 1829, Schwann, a German physiologist, made similar observations of animal tissue. Based on their observation, they put forward the cell theory in 1839. The cell theory states that all organisms are composed of one or more cells. A cell is a mass of protoplasm surrounded by a cell membrane enclosing a nucleus. Cells are basically alike in structure and function. All cells come from pre-existing cells. In 1855, Rudolf Virchow popularized the concept of omnicellula e cellula, which means all cells come from pre-existing cells. There are exceptions to the cell theory. For example, viruses do not fit into the cell theory because they don't have a cell. They have only a protein coat and a nucleic acid. Prokaryotic cells do not have a true nucleus. Instead, they have a nucleoid. The nucleoid consists of a double-stranded DNA not associated with histones. Organisms such as bread mold, or example, Rhizopus, have more than one nucleus. They have a xenocytic condition. Mature red RBC and sieve tubes of plants do not have a nucleus. Cell theory was modified, including these above exceptions. The modified cell theory is called cell principle or cell doctrine. Now let us look at the structure of a prokaryotic cell. Pro means primitive, karyon means nucleus. A prokaryotic cell is found in bacteria, cyanobacteria or blue-green algae, mycoplasma or pneumonia-like organisms, and archaebacteria. Archaebacteria are primitive bacteria. They are of three types: methanogens, which produce methane; halophiles, which are found in high salt conditions; and thermophiles, which are found in high temperature, like hydrothermal vents, hot springs, etc. Now let us look at the characteristics of prokaryotic cells. Prokaryotic cells range in size from 0.1 to 5 micrometers, and they are very small compared to eukaryotic cells. The shape of the prokaryotic cell ranges from cocci or spherical in shape, bacilli or rod-shaped, spirilla, spiral-shaped, and vibrio comma shaped. If you look at the cell of a prokaryotic versus eukaryotic, the basic difference is with regard to the nucleus. Prokaryotic cells do not have a well-defined nucleus. It is called nucleoid instead. There is no nuclear membrane. There are no membrane-bound organelles in prokaryotes, whereas in eukaryotes there are many organelles. The cell wall in prokaryotes is made up of a material called peptidoglycan, whereas in plant cells the cell wall is made of cellulose. The cellular organization of a prokaryotic cell is primitive. They lack a membrane-bound nucleus, as I told you before, and other membrane-bound organelles. The genetic material is a single double-stranded DNA called nucleoid. Histones, which are found to be associated with the DNA of eukaryotes, is absent in prokaryotes. Prokaryotic cells also lack a nucleolus and a mitotic apparatus. Mitotic apparatus consists of spindle fibers and astral rays which are formed during cell division. The cell wall of prokaryotic cell is non-cellulosic. It is made up of carbohydrates and peptides. Now let us look at the different structures of the prokaryotic cell. The outermost layer is called the glycocalyx. The glycocalyx can occur as a capsule or a slime layer. The capsule is thick and rigid and firmly attached to the cell wall. The capsule may be made of polysaccharide or polypeptide or hyaluronic acid. The ability to produce a capsule can contribute to a microbe's pathogenicity. Slime layer is loosely attached to the cell wall. It is less organized layer. It is composed of polysaccharides, glycoproteins or glycolipids. Glycocalyx allows cells to adhere to surfaces. It holds water like a sponge, preventing desiccation. Glycocalyx also protects cells from phagocytosis, which is a host defense mechanism. The major component of the cell wall is peptidoglycan or murine. It has a peptide part consisting of amino acids and a glycan part consisting of carbohydrate. It is found only in bacteria. Each layer of peptidoglycan is composed of long chains of alternating N-acetylglucosamine and N-acetylmuramic acid. These are sugars. Peptide bridges, peptides are chains of amino acids. They connect NAG and NAM with each other in the peptidoglycan layer. Here we are looking at the structure of peptidoglycan. M is denoted by N-acetylmuramic acid. G stands for N-acetylglucosamine. You can see the peptides connecting adjacent peptidoglycan. See, there are pentaglycine bridges connecting adjacent peptidoglycan. Now, gram staining. Bacteria are divided into two major groups, gram positive and gram negative, depending upon their reaction to gram staining. The gram staining method was named after its inventor, Hans Christian Gram. In gram staining, you take the preparation that you are going to observe, mix it with a drop of distilled water, then heat it and you pour few drops of crystal violet. That's a stain used to stain in bacteria. Then it is treated with iodine. Again, it is treated with alcohol. When gram-positive bacteria become violet in color, gram-positive become colorless. Then it is treated with the dye safranin. When the gram-positive bacteria will be violet in color and the gram-negative will be red in color. The main difference between gram-positive and gram-negative bacteria is with regard to the cell wall. If you look at gram-negative bacteria, the tetrapeptide chains extending from each NAM are directly cross-linked, whereas in gram-positive bacteria, these tetrapeptide chains are linked by pentaglycine cross bridges. In gram-positive bacteria, the cell wall is very thick, whereas in gram-negative bacteria, the wall is thin. In gram-negative bacteria, the cell wall is surrounded by an outer membrane that contains lipopolysaccharides and lipoproteins. Porins, which are proteins which are transmembrane, which extend from one part of the membrane to the other, allow substances to pass through the membrane. So porins are found in gram-negative bacteria. From this picture, you can make out the major differences between gram-positive and gram-negative bacteria. In gram-positive bacteria, the cell wall is very thick. In gram-negative, it is thin. You can see the outer membrane in gram-negative bacteria, which is absent in gram-positive bacteria. Another difference is with regard to the lipoteichoic acid. Lipoteichoic acid anchors the cell wall to the cell membrane, that is absent in gram-negative bacteria. In gram-negative bacteria, you can see a space called periplasmic space, which is filled with a fluid in between the outer membrane and the plasma membrane. The outer membrane contains lipopolysaccharide, which functions as an endotoxin. This endotoxin is responsible for symptoms of diseases caused by gram-negative bacteria, such as fever, hemorrhaging, and septic shock. Many antibiotic drugs, such as penicillin, are designed to interfere with peptidoglycan synthesis. They weaken the cell wall, making the bacterial cell more susceptible to the effects of osmotic pressure. Lysozyme, an enzyme found in human saliva and tears, kills bacteria by digesting the peptidoglycan in their cell walls. So what they do is they break the glycosidic bond between the two sugars in the peptidoglycan. Some bacterial species, a few archaebacteria and mycoplasma, lack cell walls. Now let us look at the plasma membrane. All cells, prokaryotic and eukaryotic, have a plasma membrane, also called cytoplasmic membrane or cell membrane or plasma lemma, that shows selective permeability, which means it allows some molecules to pass through while restricting the passage of others. There are three models of the plasma membrane. Sandwich model proposed by Danieli and Davison shows that there is a lipid bilayer in the membrane with proteins on both surfaces. These proteins were globular proteins according to them. This is the sandwich model. You can see the phospholipid bilayer surrounded by on both surfaces by the globular proteins. Second model is the unit membrane model proposed by Robertson. He proposed that the plasma membrane contained a lipid bilayer with proteins on both surfaces, just like the sandwich model. But according to him, these proteins were fibrous proteins. The most accepted model of the plasma membrane is the fluid mosaic model proposed by Singer and Nicholson. According to them, there are two layers of phospholipids with a hydrophilic head and a hydrophobic tail. Proteins are either integral proteins, also called intrinsic proteins, or peripheral proteins or extrinsic proteins. The integral proteins act as channels for the transfer of particles across the cell. The peripheral proteins function as to provide fluidity as well as mechanical support to the structure of the cell. Cholesterol is present in the eukaryotic membranes but absent in prokaryotic membranes. Instead, sterol-like compounds called hopenoids are found. But in mycoplasma, instead of hopenoids, you have cholesterol in the membrane. Let us look at the membrane transport mechanisms. One of the important functions of the plasma membrane is to control the transport of molecules into and out of the cell. The cells use various modes of transport. One is a simple diffusion, where molecules move from a higher concentration to a lower concentration. This is called passive transport also. Example, carbon dioxide may cross the membrane directly by simple diffusion. Second one is called facilitated diffusion. Charged molecules as well as large molecules need the help of carrier proteins or channels in the membrane. These structures ferry molecules across the membrane, which is called facilitated diffusion. Active transport occurs when cells move molecules across a concentration gradient. Active transport requires ATP. Therefore, active transport structures are often called pumps. Mesosomes are infoldings of the plasma membrane into the cytoplasm. A mesosome is always attached to the DNA. They may be involved in respiration, so they take the function of mitochondria in eukaryotic cells. They may be involved in DNA replication, cell wall synthesis, or secretion of enzymes outside the cell. Chromatophores. These are membranes found in the cytoplasm of photosynthetic bacteria and cyanobacteria. They are involved in photosynthesis. In cyanobacteria, granules called phycobilisomes or cyanosomes are found on the chromatophores. They contain two pigments which are together called phycobilins. The pigments are phycocyanin and phycoerythrin. Since there is more of phycocyanin, it gives a blue-green color to the cyanobacteria. Ribosomes. They were discovered by George Palade. Ribosomes of prokaryotes are smaller than those of eukaryotes. They have the 70S ribosomes. S stands for Svedberg unit or sedimentation coefficient. Eukaryotic ribosomes are of the 80S type. The 70S ribosome found in prokaryotes is made up of two subunits, 50S and 30S. The 50S subunit contains the 23S and 5S ribosomal RNA, while the 30S subunit contains the 16S ribosomal RNA. The ribosomes are actually formed of ribosomal RNA and proteins. Ribosomes are involved in protein synthesis. Sometimes many ribosomes may attach to one mRNA to form a polysome or polyribosome. Now let us look at the genetic material of prokaryotes, which is called the nucleoid or prochromosome or genophore or incipient nucleus. It consists of a circular double-stranded DNA which is not associated with histones. Remember, DNA in eukaryotes are associated with histones. There is no nuclear membrane in prokaryotes. Now let us look at plasmids. They are circular, extra-chromosomal, double-stranded circular DNA. There may be many copies of a plasmid in one bacterium. They are of two types, F factor or fertility factor, which is involved in bacterial conjugation. In conjugation, two bacteria come together attached by a conjugation tube through which the F factor of one bacterium is transferred to the next bacterium. Another type of plasmid is called the R factor, which contains genes for resistance to antibiotics. Inclusion bodies. Inclusion bodies consist of reserved food materials such as glycogen, lipid granules, volutin granules or phosphate granules, sulfur granules, cyanophycin granules, which are found in cyanobacteria, and polyhydroxybutyrate, which is biodegradable plastic. Magnetosomes. These are inclusions of iron oxide or iron sulfide surrounded by a lipid layer. They are seen in magnetic bacteria such as Magnetospirillum magnetotacticum. They help the bacteria to orient in the north-south direction. Now let us look at the photosynthetic structures. They are called chlorosomes. They are light harvesting complexes found in anoxygenic or anaerobic green bacteria. They are flattened ellipsoidal structures oppressed to the cytoplasmic phase of the cell membrane. Carboxysomes, which are found in cyanobacteria and photosynthetic bacteria, are complexes of the enzyme called Rubisco, ribulose bisphosphate carboxylase oxygenase, which is the main enzyme in photosynthesis, and another enzyme called carbonic anhydrase, which converts carbon dioxide into carbonic acid. It is surrounded by a protein coat. This structure is called a carboxysome. Now let us look at the bacterial flagellum. It is made of the protein flagellin. Bacterial flagellum has a very simple structure compared to the eukaryotic flagellum. It has only three parts, the filament, hook, and basal body. Gram-positive bacteria and gram-negative bacteria have different basal body configurations due to differences in cell wall structure. Basal body has 2 rings in gram-positive bacteria and 4 rings in gram-negative bacteria. The reason is, in gram-negative bacteria, I told you earlier, there are two membranes. One is the plasma membrane and the other is the outer membrane. So two rings of the basal body are in the plasma membrane, the other two rings are in the outer membrane. Whereas in gram-positive bacteria, only since the plasma membrane is found, it has only two rings. Based on the arrangement of flagella, bacteria are the following types: monotrichous, in which there is only a single flagellum; amphitrichous, in which there is a flagellum on either side; lophotrichous, many flagella arise from one end of the bacterium; peritrichous, in which flagella arise from the whole surface of the bacterium. Now let us look at structures called fimbriae. They are thin, bristle-like structures that are used by the cells to stick to various surfaces. Pili. Pili are longer filaments. One example is the sex pilus that holds two cells together as they transfer the DNA molecules by the process of conjugation. Now let us look at the structure of a eukaryotic cell. Example: animal cells, plant cells, and fungal cells. If you compare plants and animals, there are differences in the cells. For example, plant cells have plastids, animal cells do not. Plant cells have a cell wall, animals do not. Plant cells have a large central vacuole, animal cells have small temporary small vacuoles. Plant cells have plasmodesmata, which are cytoplasmic connections between adjacent plant cells. Animal cells do not have plasmodesmata. Plant cells do not have centrioles. Animal cells have centrioles, which are located in the centrosome. Plant cells do not have cholesterol in the membrane. Animal cells have cholesterol. The reserve food material in plant cells is starch. In animal cells, it is glycogen. See, this is the structure of an animal cell and a plant cell compared. Now let us look at detail of all the parts. The plant cell wall is a rigid and protective layer around the plasma membrane which provides mechanical support to the cell. Animal cells do not have a cell wall. They are present in most plant cells and fungi. In plant cells, the cell wall is made up of three carbohydrates: cellulose, hemicellulose, and pectin, and also proteins. Cellulose is composed of long chains of beta-D-glucose. Cellulose fibers are linear polymers of hundreds of glucose molecules. These fibers aggregate into bundles of about 40, which are called microfibrils. Microfibrils are embedded in a hydrated network of other polysaccharides. Hemicellulose is formed of branched polysaccharides. The predominant hemicellulose is xyloglucan. Pectin consists of complex polysaccharides that contain D-galacturonic acid. Proteins found in the cell wall are of two types. One is called extensin, which is a structural protein. It is a glycoprotein that contains high amount of the modified amino acid called hydroxyproline. And the second protein is called expansin, which is an enzyme involved in cell wall loosening during cell growth. Now let us look at the layers of the cell wall. The plant cell wall is multi-layered and consists of up to 3 layers. The outermost layer is called the middle lamella, then comes the primary cell wall, and then comes the secondary cell wall. While all plant cells have a middle lamella and primary cell wall, not all plant cells have a secondary cell wall. Let us look at the middle lamella. The outermost layer of the cell wall is called the middle lamella. It contains polysaccharides and pectins. Pectins aid in cell adhesion by helping the cell walls of adjacent cells to bind to one another. Then comes the primary cell wall. This layer is formed between the middle lamella and the plasma membrane in growing plant cells. It is primarily composed of cellulose microfibrils contained within a gel-like matrix of hemicellulose and pectin. The primary cell wall provides the strength and flexibility needed to allow for cell growth. Now let us look at the secondary cell wall. This layer is formed inner to the primary cell wall. It is found only in dead cells that are functional, such as xylem vessels, tracheids, etc. This rigid layer strengthens and supports the cell. In addition to cellulose and hemicellulose, it contains lignin. Secondary wall may be laid down in different layers, and in each layer, the cellulose microfibrils are arranged in different angles to increase mechanical strength. If you look at this diagram, see, the secondary wall is represented in three different layers. You can see the difference in the orientation of the microfibrils, cellulose microfibrils in each layer. This increases mechanical strength. Now, if you look at the fungal cell wall, it is a matrix of three main components. One is chitin. Chitin is a polymer of N-acetylglucosamine. Second one is glucans, which are glucose polymers that function to cross-link chitin. Third component is proteins, which are enzymes necessary for cell wall synthesis. Now let us look at plasmodesmata. These are cytoplasmic connections found in the cell wall between adjacent plant cells. The plasma membrane of one plant cell is continuous with that of the neighboring cell through the plasmodesma. The purpose of plasmodesmata involves cell-to-cell communication via transport of small molecules. The presence of plasmodesmata allows for a continuous cytoplasmic connection within the plant tissues, and it is called a symplast. The cell wall of all plant cells, which is non-living, is called the apoplast. In cells which form thick secondary cell walls, particularly the xylem vessels and tracheids, which contain no living protoplast at maturity, plasmodesmata are absent. Now let us look at another structure called desmotubule. It is a tubular connection between the smooth endoplasmic reticulum of adjacent plant cells. Now let us look at four different organelles which are always adjacent to one another, which forms the endomembrane system. The organelles are endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. First, let us look at the endoplasmic reticulum. They were discovered by Albert Cloud and Keith Porter. It is an extensive network of membrane-limited channels. ER membrane remains continuous with the plasma membrane and the nuclear membrane. It has three parts: cisternae, which are long flattened sacs; tubules, which are branch structures forming the reticular system; and vesicles, which are oval or vacuolar structures. There are two types of endoplasmic reticulum: smooth endoplasmic reticulum, which is devoid of ribosomes, and rough endoplasmic reticulum, which has ribosomes attached to it by proteins called ribophorins. Now let us look at the functions of the endoplasmic reticulum. Smooth endoplasmic reticulum is involved in the synthesis of lipids such as phospholipids, cholesterol, and steroid hormones. Rough endoplasmic reticulum is involved in the synthesis of proteins. Smooth endoplasmic reticulum is involved in detoxification of toxic substances. Endoplasmic reticulum is also involved in intercellular and intracellular transport of materials. Since it is found throughout the cytoplasm, it acts as a support for the different organelles in the cell. Next organelle is the Golgi apparatus, also known as Golgi complex or Golgi body, discovered by Camillo Golgi. Golgi bodies of plant cells are called dictyosomes. The structure of the Golgi complex is pleomorphic, which means it has different structures in different cells. But it has typically three different forms, just like the endoplasmic reticulum, that is cisternae, vesicles, and tubules. Cisternae are flattened sac-like structures arranged in bundles in parallel fashion. The concave side of the cisternae is called the cis phase or forming phase, and the convex side is called the trans phase or maturing phase. Tubules are present as tubular and branch structures that radiate from the cisternae. Vesicles are spherical bodies. Now let us look at the functions of the Golgi bodies. Number 1, glycosylation, which is the conversion of proteins and lipids produced in the endoplasmic reticulum to glycoproteins and glycolipids with the addition of carbohydrate. Number 2, synthesis of hemicellulose and pectin found in the plant cell wall. Number 3, formation of lysosomes. Number 4, packaging of proteins into membrane-bound vesicles before the vesicles are sent to their destination. Now let us look at the second structure in the endomembrane system, lysosomes. They were discovered by Christian D. Dewey. They contain hydrolytic enzymes such as amylase, protease, lipase, etc. Hydrolytic enzymes convert complex macromolecules into small molecules. For example, amylase converts starch into glucose. Protease converts proteins into amino acids. Lipase converts lipids into fatty acids and glycerol. See, now you can see in this picture the lysosomes arising from the Golgi bodies. Lysosomes are of four types. Number one, primary lysosome, which is newly formed lysosome. Number two, secondary lysosome, which is formed by the fusion of a primary lysosome with a phagosome, which is a vesicle containing solid food material, or a pinosome, vesicle containing liquid food material. During phagocytosis, which is commonly called cell eating, and pinocytosis, which is commonly called cell drinking. Number three, residual body, vesicle containing undigested food material. Number four, autophagosome. It is a vesicle formed by the fusion of a lysosome with all old or unwanted organelles. Now let us look at the functions of the lysosomes. Number one, digestion of food materials. Number two, digestion of harmful materials such as invading bacteria, viruses, etc. Number three, autolysis. This happens during cell death. During cell death, all the lysosomes in a cell break open, release their contents into the cell, which digests the whole cell. That is why lysosomes are called suicidal bags of the cell. Fourth function is autophagy, which is removal of old and unwanted organelles within the cytoplasm. Now let us look at the fourth component of the endomembrane system, vacuoles. Vacuoles are of four types. Number one, gas vacuoles, which are found in photosynthetic bacteria and cyanobacteria, which help in buoyancy. Number two, contractile vacuoles, which are found in protozoa like amoeba or paramecium. They help in osmoregulation or maintenance of the fluid balance of the body and in excretion. Third, food vacuoles or secondary lysosomes, which are formed during phagocytosis and pinocytosis, which help in digestion of food materials. A food vacuole is formed by the fusion of a primary lysosome with a phagosome or a pinosome. Fourth, vacuoles are sap vacuoles, which are found in plant cells. Plant cells generally have one large vacuole that takes up most of the cell's volume. A selectively permeable membrane called the chromoplast surrounds the vacuole. The fluid inside the vacuole is called the cell sap, which consists of water, minerals, sugars, and organic acids, and red pigments called anthocyanins. Now let us look at ribosomes. Ribosomes are the smallest organelles. They do not have a membrane surrounding them. They are made of ribosomal RNA and protein. They are synthesized in the nucleolus. In this picture, you can see ribosomes attached to the endoplasmic reticulum. Eukaryotic ribosomes are of the 80S type, comprising of two subunits, 40S and 60S. The smaller 40S subunit consists of one molecule of 18S ribosomal RNA and 30 proteins named as S1, S2, S3, and so on. The larger 60S ribosomal subunit consists of three types of ribosomal RNA, 28S, 5.8S, and 5S ribosomal RNA, and 40 different proteins which are named as L1, L2, L3, and so on. Some of the ribosomes are attached to the endoplasmic reticulum, others are free in the cytoplasm. The free ribosomes synthesize proteins for internal use, cytoplasmic proteins and enzymes. Those which are attached to the endoplasmic reticulum synthesize proteins for external use, example, secretory or lysosomal enzymes. The ribosomes which are seen in chloroplast and mitochondria of eukaryotes are of the 70S type, which is similar to the ribosomes found in prokaryotes. Now let us look at plastids and mitochondria. Let us look at the similarities. Both of them have a double membrane. Both of them have circular DNA and 7ds ribosomes, which are prokaryotic characters. Both of them are endosymbionts, which means originally they were bacteria which were engulfed by the eukaryotic cell. Now they are living in symbiosis with the eukaryotic cell. They are semi-autonomous organelles, which means even though they have their own DNA, they are under the control of nuclear DNA. Another similarity is both of them synthesize ATP. But ATP synthesis in the mitochondria is called oxidative phosphorylation, whereas ATP synthesis in the chloroplast is called photophosphorylation. Now let us look at plastids. Plastids were discovered by Ernst Haeckel. They are of three types: chloroplast, chromoplast, and leucoplast. First, let us look at chloroplasts. They have a double membrane. They have stack of membranes called thylakoids, which form a granum, stroma, which is a fluid inside with a circular DNA and 7ds ribosomes, and grana are connected by stroma lamellae, also called frets. Chloroplast is a site of photosynthesis. The pigments of photosynthesis, namely chlorophyll A, chlorophyll B, carotene, and xanthophyll, occur in groups called photosystems, which are located on the thylakoid membrane. The light reaction of photosynthesis occurs on the grana of the chloroplast. Dark reaction, also called Calvin's cycle, occurs in the stroma of the chloroplast. Now let us look at the chromoplasts. They arise from chloroplasts by the breakdown of chlorophylls. They contain pigments called carotenoids, which are a combination of carotene and xanthophyll. When they are present in flowers, they help in pollination. When they are present in fruits, they help in the dispersal of fruits and seeds. The third plastid is called leucoplasts. Leucoplasts are colorless plastids without grana. They are found in underground storage organs. They are of three types: amyloplasts, which store starch; aleuroplasts, which store protein; and elaioplasts, which store oil. Now let us look at mitochondria. Mitochondria were observed for the first time by Kolliker, and he called them sarcosomes. The name mitochondria was given by Benda. Mitochondrion is surrounded by a double membrane. The inner membrane produces finger-like projections called cristae. The fluid inside the mitochondrion is called mitochondrial matrix. Oxysomes, or F0, F1 particles, or ATP synthesis, are enzymes which synthesize ATP, which are located on the inner membrane. In the mitochondrial matrix, there is a circular DNA and 7ds ribosomes. Now let us look at the functions of the mitochondria. Three phases of cellular respiration occur in the mitochondria. Krebs cycle occurs in the mitochondrial matrix. Electron transport occurs in the inner membrane of the mitochondria. ATP synthesis occurs on enzymes called ATP synthesis. The most important function of mitochondria is ATP synthesis. That is why they are called powerhouses of the cell. The synthesis of ATP in mitochondria is called oxidative phosphorylation. Now let us look at how ATP is synthesized. Chemiosmotic theory, proposed by Peter Mitchell, explains how ATP is synthesized. The basis of this theory is the creation of a proton gradient. During cellular respiration, NADH and FADH2, produced during glycolysis and Krebs cycle, pass electrons through electron carriers located at the inner mitochondrial membrane. They usually belong to a group of proteins called cytochromes. Protons are transferred from the matrix to the inner membrane space during electron transport. This results in a proton gradient. ATP is synthesized when the protons move back to the matrix through channels in ATP synthesis. The energy in the protons is used to combine adenosine diphosphate and inorganic phosphate to form ATP. Now let us look at two small organelles which are called microbodies. They are single membrane bound organelles. They are of two types, glyoxysomes and peroxisomes. Glyoxysomes were discovered by hairy beavers. They are abundant in oil-rich seeds. They are the sites of glyoxylate cycle in which fats are converted into glucose during the germination of these seeds. Peroxisomes were discovered by Christian D. Dewey. Certain peroxide forming enzymes and catalase that are involved in the formation and degradation of hydrogen peroxide are found in peroxisomes. In plants, they are involved in photorespiration, which occurs due to the oxygenase activity of the enzyme Rubisco. Photorespiration occurs in three organelles: chloroplast, peroxisome, and mitochondrion. In animals, they are involved in beta oxidation of fats, in which fats are converted into acetyl-CoA, which enters the Krebs cycle. Now, let us look at the cytoskeletal structures. These are protein fibers present in the cytoplasm that give shape to the cell and support various organelles. They are of three types: microtubules, microfilaments, and intermediate filaments. If you look at this picture, you can see the three cytoskeletal structures. They are found throughout the cytoplasm. Microtubules were discovered by D. Robertus and Franchi. They are hollow cylindrical structures, the wall of which is formed of 13 protofilaments. Each protofilament is formed of the protein tubulin. They are components of cilia, flagella, centrioles, which are found in the centrosome, spindle fibers, and astral rays. Together they are called mitotic apparatus formed during cell division. Microfilaments were discovered by Pelvis and others. Microfilaments are also called actin filaments. They are polymers of the protein actin. They are present in muscle cells where they form myofibrils. They are also present in the microvilli of the intestine. They cause cyclosis, which is streaming movement of the cytoplasm. They produce contraction of muscles. They produce movement of microvilli. They help in the process of cytokinesis, which is division of the cytoplasm, in which the cell pinches off and physically separates the two daughter cells. Intermediate filaments were discovered by William Asbury. They are intermediate in size between microtubules and microfilaments. They have been classified according to the constituent proteins such as desmin filaments, keratin filaments, vimentin filaments, lamin filaments, etc. Besides cytoplasm, they are also found in the inner nuclear membrane to form the nuclear lamina. The chromatin fibers in the nucleus are attached to the nuclear lamina. Centrosome, discovered by Van Beneden. It occurs in animal cells and those plant cells which have cilia and flagella, such as the green alga Chlamydomonas and male gametes of bryophytes, pteridophytes, and gymnosperms, which are multiciliate. Centrosome contains two cylindrical structures placed at right angles to one another called centrioles. A pair of centrioles is called a diplosome. There is no membrane surrounding the centrioles. There is a dense mass of cytoplasm surrounding the centrioles which is called pericentriolar material or maculae or MTOC, microtubule organizing center. It is called MTOC because it is the place of origin of all the microtubules in the cell. Each centriole of the centrosome contains 9 triplets of microtubules arranged in a cartwheel fashion. There is a hub in the middle from which 9 spokes arise and 3 microtubules are attached to each spoke. This kind of arrangement is called 9 plus 0 arrangement. It is called 9 plus 0 because when compared to cilia and flagella, the 2 inner microtubules are absent here. Centrosome forms spindle fibers and astral rays during cell division. Centrosome forms the basal body of cilia and flagella, which also shows 9+0 arrangement. Now let us look at cilia and flagella. They are tiny hair-like projections from the cell made of microtubules and covered by the plasma membrane. Cilia are shorter than flagella. Cilia are present in much greater numbers, but there is only a single flagellum per cell. They differ in the nature of movement also. In the case of flagella, the movement is whiplash type of movement. In the case of cilia, it is usually circular movement. Structurally, both cilia and flagella are similar. There is a bundle of microtubules called the axoneme in the middle, surrounded by the membrane that is part of the plasma membrane. The axoneme is connected with the basal body, which originates from the centrioles. The septum, which separates the basal body and the upper part of the cilium or flagellum, is called the basal plate. If you take a section of the cilium or flagellum, above the basal plate, it shows 9+2 arrangement, but the basal body shows 9+0 arrangement. The two microtubules are absent in the basal body. Each axoneme is filled with a matrix in which are embedded two central microtubules and 9 outer pairs of microtubules called doublets. Like the centriole, there is a hub in the middle from which 9 spokes arise. The outer microtubules are attached to the spokes. Arising from the outer microtubules, there are structures which are called dynein arms. Dynein arms contain ATP synthase, which is responsible for the production of energy for the movement of cilia and flagella. The outer microtubules are connected by a protein called nexin. Now let us look at the nucleus. Nucleus was discovered by Robert Brown. The nucleus carries the genetic information necessary to regulate the different cell functions. So it is the most important organelle in a cell. It consists primarily of DNA, about 20% of its mass, DNA binding proteins called histones, and some RNA. You can see that the outer membrane of the nucleus, which is the nuclear membrane, is continuous with the endoplasmic reticulum. The nuclear membrane is a double membrane with numerous nuclear pores. Surrounding the nuclear pore, there are two annuli, an outer and an inner annulus, each with 8 globular subunits. The complex formed of the nuclear pore and the annuli is called the pore complex. Nuclear lamina consists of intermediate filaments on the inner nuclear membrane to which chromatin fibers are attached. The fluid inside the nucleus is called nucleoplasm. It contains DNA polymerase, the enzyme which is involved in DNA replication, RNA polymerase, which is the enzyme involved in transcription, that is the formation of RNA from DNA, and 4 types of nucleotides that form DNA and RNA. A nucleotide has 3 parts: nitrogen base, sugar, and phosphate. Nucleolus, these are spherical structures which are 1 to 4 in number, which are the sites of ribosome synthesis. Ribosomes are synthesized by combining ribosomal RNA and protein. The most important structure inside the nucleus is the chromatin fibers, which are fibers formed of DNA and histones. Now let us look at the structure of the chromatin fibers in detail. Under the electron microscope, chromatin fibers show bead-like structures called nucleosomes. Each nucleosome consists of 8 histones on which DNA is wound. Adjacent nucleosomes are connected by linker DNA. Histones are of 5 types: H1, H2A, H2B, H3, and H4. In a nucleosome, there are 2 each of H2A, H2B, H3, and H4. H1 does not occur in the nucleosome. Instead, it sits on the top of the structure, keeping in place the DNA that has wrapped around the nucleosome. If you look at the picture, you can see 8 histones, 2 each of the 4 different types of histones. And you can see the location of the H1 histone. Solenoid. A solenoid is a spirally coiled structure in which there are 6 nucleosomes in one coil. All the H1 histones of the nucleosomes are projecting to the center. The chromatin fibers occur in the nucleus in the form of a solenoid. Chromosome. Chromosomes were discovered by Strasburger. During cell division, the chromatin fibers, or we can say the solenoid, are converted into chromosomes by further coiling. From this picture, you can make out the different types of coiling found in the chromosome. Now, if you look at the structure of a chromosome is surrounded by a protein sheath called the pellicle. It has a centromere or primary constriction. On the centromere, there are protein discs called kinetochores to which spindle fibers are attached. There are 2 chromatin fibers or chromonemata inside the chromosome. Another structure is the secondary constriction or nucleolar organizer. That is the place of origin of the ribosome. So, from the secondary constriction region, ribosomal RNA is produced, which combines with protein to form ribosome. The tip of the chromosome is called a telomere. Human telomere consists of 2000 repeats of a sequence TTA-GGG. The length of the telomere is related to aging. The telomere becomes shorter and shorter as you age. Telomere was discovered by Elizabeth Blackburn, who received Nobel Prize for this discovery. Depending upon the position of the centromere, chromosomes are of the following types. Metacentric, in which centromere is exactly in the middle. Submetacentric, where the centromere is towards one side. Acrocentric, where the centromere is near the end. And telocentric, where the centromere is at the end. Now let us look at special types of chromosomes called giant chromosomes. These chromosomes show a phenomenon called endomitosis, in which the chromatids in a chromosome keep on dividing, but the chromosome does not divide. So finally, the chromosome ends up with many different chromatids. They are of 2 types: salivary gland chromosomes or polytene chromosomes and lamb brush chromosomes. Salivary gland chromosomes or polytene chromosomes were discovered by Balbiani. They are present in the salivary gland cells of larvae of insects such as Drosophila. The arms of the chromosomes are fused at the center to form chromocenter. This is a picture of the salivary gland chromosomes of the fruit fly Drosophila. You can see the chromocenter in the middle, which is formed by the fusion of 8 chromosomes found in the cell of Drosophila. The arms of the chromosomes show dark bands and light bands. The dark bands are heterochromatic regions in which the DNA is tightly coiled. Some of the dark bands temporarily swell up and form enlargements called chromosome puffs or Balbiani rings. These regions actually represent uncoiled DNA where active transcription or synthesis of RNA occurs. Now let us look at lamb brush chromosomes. They were discovered by Fleming. These chromosomes occur in the oocytes of amphibians and in some insects. They are extremely large homologous chromosomes which can be seen in the diploidine stage of prophase 1 in meiosis. A lamb brush chromosome has an axis from which paired loops extend in opposite directions, giving the appearance of a lamb brush. The axis consists of bead-like structures of chromomeres, which are actually nucleosomes, and interchromomere regions. The loops consist of uncoiled DNA where active mRNA synthesis occurs. Thank you for listening to my